The following presentation is on the use of DNA fingerprinting in forensics. The presenters for this presentation are myself, Hoyda Bristow, Fern Rodriguez, and Aurelia Bello. The following elements will be discussed. What is the purpose of DNA fingerprinting? What's it used for? The history and development, how it works, current applications, advantages and disadvantages, and potential future applications. So what is DNA fingerprinting? Well, Lutz describes it as a comparison of the DNA in a person's nucleated cells with that identified in biological matter found at the scene of a crime or with the DNA of another person for the purpose of identification or exclusion. So I'm briefly going to give a basic explanation of DNA fingerprinting, which Fern will later expand on this, on the complexities of DNA fingerprinting. So basically our DNA is made up of over 3 billion nucleotides, which is 99% of this is the same from one person to the next. So what DNA printing does, it looks at the 0.1% that isn't the same. So to put that another way, if each nucleotide was 2.5 centimeters in length, the nuclear DNA in each cell would be more than 160,000 kilometers long. But DNA fingerprinting only looks at about 90 meters of this information. So that's a tiny amount that they're looking at. DNA fingerprinting, it can be used by forensic scientists to determine the identity of a person by blood, tissue, or hair follicles to high levels of accuracy. So the main types of DNA fingerprinting methods in use are restriction fragment length polymorphism, or RFLP, polymerase chain reaction, PCR, amplified fragment length polymorphism, AMFLP, and short tandem repeats, STR. And I'll briefly go over each one. So polymerase chain reactions, this can be broken up into 7 stages. So DNA is extracted from the cells. It is then cut up using an enzyme, a restricted enzyme. It's separated into fragments onto a gel. It is then transferred onto paper. A radioactive probe is then added to determine DNA fragments, and finally an X-ray film is developed. And as I discussed before, Fern will go into a deeper level, more complex explanation about PCR. So the next method is restriction fragment length polymorphism, or RFLP. This is used for analysis of unique patterns in DNA fragments, differentiate between organisms. This requires large amounts of DNA and it cannot be degraded. Now we have amplified fragment length polymorphism. Okay, this method only involves small amounts of DNA. Its restricted fragments are amplified, reducing the complexity of material to be analyzed, and it works on a variety of genomic DNA samples. And the fourth method is short tandem repeats, or STR. This requires larger amounts of DNA. It's less susceptible to degradation, and it can be recovered from bodies or stains that have decomposed. So those are the methods used in DNA fingerprinting. So what is the purpose of DNA fingerprinting in forensics? Well, in forensics, it assists police in criminal investigations and helps convict criminals or exonerate wrongly accused. It helps scientists can determine whether 2 samples of genetic material are from a particular individual. Now, DNA fingerprinting in forensic science, as a standard for use in criminal investigations, paternity testing is relatively recent. Before DNA fingerprinting was discovered, new markers for identity were based on variations of serum, proteins, and red blood cell enzymes. Eventually, the human leukocyte antigen system was used. Sir Alec Jeffreys. So in it was only in 1985 when this man, Sir Alec Jeffreys, a geneticist from the University of Leicester, discovered a technique which identifies an individual based on unique patterns from variations in DNA. In other words, his discovery demonstrated a single test could distinguish everyone on Earth except for identical twins. The impact of DNA fingerprinting on criminal investigations and individual investigations cannot be understated. It revolutionized forensic investigations as we know it. However, Sir Alec Jeffreys' discovery would not have been possible without the groundwork done by early pioneers in forensic science. Many have helped shape DNA fingerprinting in forensics to where it is today. However, I selected these individuals who are key historical figures in the development of DNA fingerprinting. Matteo Orfila. So he was known as the father of toxicology, and he was a chemist and toxicologist, and at the time he was dissatisfied with the many inadequacies and accuracies of forensic chemical tests. The primary type of poison at that time was arsenic, but there was no really reliable ways of testing for its presence. Orfila created new techniques which demonstrated effects of poisons on organs and tissue. This is still used today in forensics where a murder may have been committed using poison. Orfila is also credited as one of the first to use a microscope to assess stains of blood and bodily fluids. His work refined forensics as a science. Hans Gross has been called the father of criminology. He applied science to the practice of crime investigation. He studied offender behaviour and other methods for profiling murderers, thieves and other criminals. He was an advocate of professional ethics, objectivity and inspection of evidence. Edward Locard was called the father of the crime lab. He incorporated the scientific method and trace evidence analysis for crime scene investigation and reconstruction of a crime. He believed the criminal would leave some trace at the crime scene and take some away. This is called the Locard's exchange principle. He used techniques such as hair fiber and also dust analysis. Carl Landsteiner noted that blood serum from some individuals would agglutinate or clump up the red cells of other individuals. He discovered that human blood could be classified into 4 general types, A, B, O, or AB. This discovery paved the way for blood transfusions to be carried out safely and won him the Nobel Peace Prize in 1930. Leon Lattes, he devised a procedure for determining the blood type AB, or O of a dried blood stain. He applied Landsteiner's ABO blood group technique, and this method, he immediately applied to criminal investigations for identifications of an individual.
Now we will briefly talk about where DNA fingerprinting is used and a little more deeply about how it works. DNA fingerprinting is a technique that can be used to investigate biological relationship between people, to identify organisms like bacteria strands in cheese, infections control, and plant genotyping, just to name a few. And also to help solve crimes, which is the focus in this presentation. Forensic science relies on DNA analysis' reputation for being the ultimate method of biological individualization. Forensic geneticists are not interested in the entire genome of individuals. Their primary interest is in a much smaller number of highly variable regions of the DNA, which is just about 1/1,000,000th of the genome. Also, forensic analysis favors DNA sequences that are external to the genes and are not believed to encode biological traits. The techniques used in this field exploit those places in the genome known as polymorphic loci, which exhibit detectable variations. and these variations can be in terms of the sequence of DNA bases or in terms of length. But how does DNA fingerprint work? To explain how DNA fingerprinting is used, let me briefly talk about the differences in length of specific regions in our DNA that result from unequal crossing over within these regions during the development of sperm and new oocyte precursors and also during meiosis. As a consequence of this unequal crossing over, the lengths of the specific DNA regions are unique in each person, a fingerprint in your DNA. In 1986, Carrie Banks Mullis developed a method that replicates or amplifies a DNA sequence that is from 100 to 2,000 base pairs long, producing millions of copies of the initial DNA sequence through a chain reaction in which the products of one cycle of replication become the templates for the next. That is known as polymerase chain reaction, or PCR, and he was awarded a Nobel Prize for it in 1993. So what forensics geneticists do is to use PCR to replicate relatively small sequences of someone's DNA in order to compare it with a marker, which is a known DNA sequence, based on the length of these sequences, what is called short tandem repeats. Short tandem repeats are short tandemly repeated DNA sequences that involve a repetitive unit of a certain number of base pairs. Within the same species, each individual will present a different number of repeats with consequent different lengths of these STRs, a DNA fingerprint. The principle behind the PCR technique is quite simple. It starts with isolating from someone's tissue a small DNA sequence, which is our target sequence. Then the target sequence, which will be the template for the reaction, is placed in a solution with synthetic primers, which are short pieces of single-stranded DNA that are complementary to the target sequence. In the solution also goes a heat-stable DNA polymerase and additional selected nucleotides. Then the first cycle starts. The solution is heated up to 94-98 degrees Celsius, which denatures the double helix DNA strand, breaking its hydrogen bonds and releasing the two single strands with their now exposed nucleotide sites. The temperature is then brought down to 50-58 degrees, which allows the synthetic primers to form hydrogen bonds with their matching sequences on both 5' and 3' ends of the template sequence, which limits the sequence to be replicated, a step that is called primer annealing. After that, the system is again heated up, this time to 70-80 degrees, and elongation or primer extension starts. Elongation is the DNA synthesis per se. And this happens due to the presence of the heat-stable DNA polymerase that catalyzes the binding of new nucleotides onto the template strand following the coding initiated by the primers. The cycle is then repeated up to 30 times, and because the products of one cycle of replication become the templates for the next, the replication is exponential in every cycle, producing millions, even billions new target sequences or the short tandem repeats in the end of the process. This is why PCR is said to amplify a DNA sequence. The whole process usually takes just a couple of hours. After 3 cycles, the target DNA sequence defined by the primers begin to accumulate. As more and more units of the target sequence are produced, flanking regions are also produced. But they are not part of the target sequence and will not be used for detection later on. But since the number of target sequence amplifies exponentially in each cycle, after 20-30 cycles, the system will contain enough amounts of it for detection. To identify and compare DNA sequence sizes from different samples, geneticists use electrophoresis. Electrophoresis is based on molecule size and charge. We know that phosphate molecules that make up the backbone of DNA molecules have a high negative charge, and when the sample is placed in a buffer solution which an electric current runs through, it is drawn to the positive end of the system. Small DNA sequences will travel further, closer to the positive electrode, and the larger ones will stop somewhere behind. The stain they leave on the gel forms the bands we see on the screen. Have I said gel? What gel? Ah, that gel. In the beginning, the material used to be loaded onto an agarose gel matrix, then stained and analyzed. The gel was then dried and stored for future reference. Quite troublesome. The picture on the top right is an autoradiograph, which is an X-ray of the agarose gel matrix. And since the late 1990s, it has improved as STR multiplexing kits, and they have combined PCR and labeling through the use of fluorescent primers. And more recently, PCR products have been separated in liquid phase by capillary electrophoresis rather than on solid gel, allowing many more samples to be separated and analyzed in a single run. The fluorescently labeled PCR fragments are detected by laser as they travel past a fixed point under electrophoresis, and a computer software then measures the size of fragments by comparison and represents the profile in various ways, including a graph called electropherogram.
Now I'll talk to you a little bit about the advantages, disadvantages, and the future applications of forensic DNA fingerprinting. A big advantage that we're all probably aware of is the fact that DNA fingerprints help to solve crimes. It can convict criminals, exonerate the wrongly accused, help victims of disasters and wars, as well as establish paternity in child support litigations and custody. It's practically the key to linking suspects to biological evidence. Another advantage is that DNA testing can be applied to any human nuclei containing cells. So these can include saliva, semen, urine, and hair. DNA fingerprinting is also used to prevent and diagnose inherited disorders and certain types of diseases. Interestingly, I'm going to use an example in which DNA fingerprinting is used in the prevention of Mycobacterium tuberculosis. This use of DNA fingerprinting is relatively a new laboratory technique and was originally established in 1996 by the Centers for Disease Control and Prevention. It was a 5-year prospective population-based study which was conducted from 1996 to 2000. Another great benefit of DNA fingerprinting is that it is actually able to resist degeneration even after exposure to chemicals and bacteria. DNA fingerprinting is one of the greatest discoveries in the 20th century, which has actually revolutionized the science of forensics. There are also some disadvantages which are worthy to mention. So we can have errors such as cross-contamination, and DNA fingerprints can actually be easily planted at the crime scenes. False negatives or false positive results can be present with the older DNA profiling technologies. So I'll use an example, a false negative. We could have a case where a person is acquitted for a crime that they in fact did commit and they did have their DNA at the crime scene. So the criminal is guilty. But if they had their DNA run through an older profiling technology, this may be able to provide this due to some limitations and therefore falsely coming to a conclusion that the criminal is not guilty, creating a false negative. This can also be applied vice versa if a false positive were to occur in the situation. We need to take into consideration that with DNA fingerprinting, only statistical probability is possible. This means that figures with 100% absolute certainty can never be established. However, the more people that are tested, the lower the statistical probability. The last two disadvantages can be applied together or in addition to one another. So, as we're all aware of now, computer hackers are an actual reality. Stored DNA information in databases are prone to hackers, and this can exploit someone's genetical information. This is such a worry, as our genetical information is inherently identifiable and can be put to misuse very easily. In addition to this, of course, exploitation is a big violation of someone's privacy. But if we look into the fact that innocent people are sometimes required to give their DNA information unwillingly for investigation, it is then uploaded to databases, and in some cases it can be deleted, but other times it stays in the system. A few people believe the principle innocent until proven guilty, so that sort of also applies here. So people who are not convicted of a crime may still have their database in the systems. In which direction will the future of DNA fingerprinting be heading? That's the same question the forensic community asks. So in this slide, we'll be talking about an article by Butler in 2015, which talks in depth about the future of forensic DNA fingerprinting and analysis. Let's start off with faster results. DNA testing protocols can be expected to become much faster with rapid DNA instrumentation. The rapid DNA instrumentation combines together the steps of DNA extraction, rapid DNA PCR, DNA separation, detection, size, and genotyping. The initial versions of these rapid DNA systems can provide swab-in to profile out results in less than 90 minutes for each reference sample. In order for rapid DNA technology and devices to reach their full potential, more database systems will be needed to be implemented, but not at the risk or sacrifice of quality. So the future needs quality as well as speed. Higher sensitivity and information content. So in the future, there will be higher information content and detective sensitivity. Even throughout the years, we have seen the development of higher amounts of data to be recovered from biological evidence. The future of more powerful information in DNA analysis can include better detection sensitivity and higher information content from core STR loci, genetical markers, and higher information from sequence analysis. Stronger conclusions. There are some DNA samples that are more challenging than others. These can include poor quality samples which may contain sorts of degraded DNA, PCR, or inhibitors that fail to produce the informative profile. These will require a software or database that can understand the limitations and therefore produce stronger conclusions with probabilistic approaches of the complex evidence. Some other factors which can be present in the potential future applications of DNA fingerprinting can include moving forward with evidence interpretation and creating more cost-effective economy scales. So, in conclusion, there are many hopes for future applications in DNA fingerprinting, and with science growing so rapidly, we are hopefully able to get there soon. Thank you. Before we go, we would like to acknowledge the achievements of Mr. Kohara, who in 1986 shared a Nobel Prize with his colleagues for a research that helped to show how the nucleotides in nucleic acids control the cell synthesis of proteins. This week it would have been his 96th birthday. Thank you for watching.